Hello, everyone. My name is Tyne Lee, and I will present our multitask matrix factorization method to detect structural changes in 3D genome organization. So 3D genome organization refers to how the DNA is folded in a systematic way inside of the cell's nucleus. And distant regulatory regions like enhancers and promoters can be brought together in 3D space, triggering gene expression and influencing downstream cellular processes. And we can study this 3D genome organization with high-C or high-throughput chromosomal conformation capture technology. High-C measures the tendency or the level of interactions between a pair of genomic regions, including enhancers and promoters. So it generates a symmetric matrix like the one you see on the right, whose entry represents how often the region corresponding to the row interacts with the region corresponding to the column. And high-C data reveals how the 3D genome is organized into different structural units. And the unit of interest to us are TADs, or topologically associating domains. TADs are visible as these blocks along the diagonal of high-C matrices, and they represent groups of highly interacting neighboring genomic regions. And their boundaries are enforced by various architectural proteins that bind to the DNA and insulate regions in one TAD from a neighboring one. And we're in particular interested in identifying shifting TAD boundaries or dynamic TAD boundaries because of their role in development and in disease. So here, just as an example, we see an inversion around the TAD boundary, which causes it to shift, which in turn creates a deviation in this enhancer-driven gene regulation of WANT6 gene, leading to a developmental disease, in this case, F syndrome syndactyly. So a key challenge in regulatory genomics is to pinpoint such regions that are involved in these TAD scale changes so we can ultimately link them to downstream consequences. To this end, we've developed a pipeline that involves a multitask matrix factorization framework with some downstream annotation tasks, which we will walk through now. The core component of our proposed method is non-negative matrix factorization, or NMF. So just as you can factor a number into two smaller numbers, you can factor a large matrix into two smaller matrices. And these smaller factor matrices can be used to co-cluster the row entities to the column entities. So as an example here, we have a toy Netflix challenge example where we're given a matrix of user movie ratings. And let's assume there are two broad groups of users, so the green group and the purple group, that are broadly interested in two different genres of movies, the green genre and the purple genre. And NMF allows us to identify these groups from the lower dimensional matrices as depicted by this picture here. So formally, the goal of NMF is to find these two lower dimensional factors, U and V, that will approximate the input matrix X when they're multiplied together. So we extended this to a multitask framework called TGIF, a tree-guided integrated factorization, so we can naturally compare across multiple contexts for a particular use case. So in our proposed method called TGIF, each task corresponds to a biological condition or a cell line or a cellular state and has its own distinct input high-C matrix. For each task T, our goal is to find task-specific U's and factors U's and V's that will approximate the input matrix X. And the factors from tasks that are closely related in the task tree will be constrained to be more similar to their shared parent factor, as enforced by this regularization term here. This tree structure specified by the user to encode the task relationship, it allows us to handle data sets with arbitrary relatedness and to flexibly capture any task heterogeneity. And to learn these factors that optimize this objective, we use a type of block coordinate descent algorithm with a guarantee to a local minimum. So what do we do once we have these factors? From these lower dimensional factors, we want to quantify TAD boundary level changes. That's our ultimate goal. So first, we calculate boundary scores for each region. So if you imagine this is the length of the chromosome, the boundary score for region I is its cosine distance to its immediate upstream neighbor I minus 1 in this lower dimensional space. So you kind of see this peak in the boundary score here. And since we don't know how many TADs or boundaries are in a given chromosome, this is an unsupervised learning problem, we sweep the hyperparameter K and take the mean boundary score across all these Ks for each genomic region for each task. And we compare the mean boundary scores against this background score set that is generated from a randomly shuffled matrix. This allows us to calculate each boundary score's empirical p-value and determine their significance. So what we end up with is a list of significant boundaries within each task. And finally, once we have identified these significant boundaries, we can then annotate each region for their context-specific boundariness or for their persistence and boundariness across all these different tasks. So here we show, as an example, a final output bed file containing such annotations for each of our genomic regions. To test the effectiveness of TGIF, we benchmarked it against two other approaches using pairs of simulated data sets with known ground truth boundary differences. And we use the simulation procedure from Furcato et al. We find that TGIF in pink identifies fewer false positive differential boundaries, leading to overall higher F-score. And this holds true for varying degrees of differences between the pair of simulated data sets. And TGIF can also recover with fairly high accuracy, according to the F-score, a common or shared set of boundaries between the pair of tasks. And we next apply TGIF to a real Hi-C data set. So this is Hi-C data sets captured during cardiomyocyte differentiation at 6 different time points, starting at human embryonic stem cell, differentiating all the way to ventricular cardiomyocytes. Here's an overview of the number of boundaries identified by TGIF. Around 21,000 boundaries are found in the union of boundaries across all time points, and about 1/10 of them are in the intersection of boundaries, so these are the shared common boundaries. Within each time point, we identified about 8,000 to 10,000 boundaries across all chromosomes, and about 1/9 of them are only found within each time point, so the red bars are the unique boundaries. We also measure the similarity or the overlap of boundary sets between pairs of time points using Jacquard index. So a higher Jacquard index corresponds to a larger overlap in the boundary set between a pair of time points. And we see that adjacent time points share more boundaries as expected, and the fully differentiated cardiomyocytes at day 80 have the most distinct boundary sets. Zhang et al., who had originally used this data set to show that a particular retrotranspose called ERV-H, when transcriptionally active, demarcates TAD boundaries in human pluripotent cells. So here you see at day 0 in stem cells before differentiation, there's this transcriptionally active ERV-H locus that forms a very strong TAD boundary, which dilutes out as differentiation proceeds in the subsequent days. Validating this point, when we look at the top 100 transcriptionally active ERV-H sites, we find higher TGIF boundary scores at day 0 pluripotent state than in any other subsequent time points during differentiation. And here we just show as an example one such top transcriptionally active ERV-H site that coincides with a significant TGIF boundary at day 0, but this disappears in subsequent time points as expected. And I'm only showing the first 3 time points here, but the boundary also disappears in the subsequent time points as well. Now, we next use TGIF to identify potential novel context-specific boundary elements or architectural proteins. And the way we did that, we used these motif match sites of different transcription factors and intersected them with ataxic peaks and used it as a proxy for their actual binding in a context or cell line of interest. In our case, it's human embryonic stem cells. So this approach allows us to sweep more than 700 transcription factor motifs in the Jasper database, even if they actually don't have this ChIP-seq binding data set in our cell line of interest. So we looked for transcription factors whose accessible motif sites are enriched in these HESC unique boundaries that were identified by TGIF. And here are the top 10 transcription factors by their accessible motif site enrichment in HESC unique boundaries. CTCF pulls up at the top. It is a well-known TAD boundary enforcing element. YY1, together with CTCF, is known to play a role in early development and pluripotency. And MYC is also known to play a role in chromatin architecture as well as enhancer looping. So all these other transcription factors might be of interest for further investigation. Finally, we started to analyze disease-associated SNPs and their relationship with differential or shared boundaries that are identified by TGIF. So we looked at around 4,800 GWAS SNPs associated with cardiovascular disease in the GWAS catalog, and found that about 9% are found in a boundary in at least one of the time points during the cardiomyocyte differentiation. So these are TGIF boundaries. And interestingly enough, we found that when compared to all genomic regions, TGIF boundaries tend to be significantly depleted for such disease-associated SNPs. So here on the right, we are showing an example region with HESC unique boundary containing one such SNP, and the boundary disappears in subsequent time points. So such regions could be further investigated to link non-coding SNPs to their specific function in local chromatin organization. So in conclusion, TGIF is a multitask matrix factorization method to identify regions involved in a dynamic 3D genome organization. It effectively recovers ground truth TAD differential boundaries in simulated data. And when we applied it to real high data from cardiomyocyte differentiation, we were able to demonstrate that it can be used to identify context-specific boundary elements and those potentially involved in disease-associated SNPs. So I'd like to thank my advisor, Sushmita, and the members of the Roy Lab. We're able to do our research thanks to the funding from the NIH and the CIBM training program, as well as the computing infrastructure that's maintained by the Center for the High Throughput Computing. And we'd also like to thank Yanjiao Zhang and Bing Ren for providing us with the list of transcriptionally active ERV-H sites during the cardiomyocyte differentiation. And I think I still have time. So here's a plug for the biomedical data science graduate program that I'm part of. There will be an open house for prospective grad students online on December 5th. If you have any questions about the program, about grad school life, please feel free to talk to me or reach out to Sushmita Roy, who's also the director of the PhD program. Thank you for listening.
Any questions?
Hi, very nice talk.
Thank you.
I had two questions. So how much of an impact does that NMF reduction have in reducing that noise in your boundary detection? And then the second question is, when you have a time series like day 0 to day 5, what kind of a tree structure can you get? And does that tree depend on the gene expression of these states or the high C of these states? Where do you get the tree?
Those are really good questions. So the first question was, how does NMF help with dealing with the noise presented in these high data sets? And high data sets are very noisy and often very sparse, especially depending on the depth. So we had actually published another method called Grinch before this one, where we basically apply a single task NMF with some smoothing capability to actually handle noise. And we do find that NMF is in fact very good about handling a lot of the noise, especially at the lower resolution high data set, but also at the higher resolution, even noisier, sparser high data set as well. So basically condensing the data helps remove a lot of the noise that is present within the high data set. That's what we at least find. And then the second question was, the tree structure. How do you define the tree structure? With the time series one, we basically just kept adding branches as there were subsequent time points. So this kind of this stacked branch as we went to the subsequent time point. Another way you can just do it when you're not sure about the tree structure is just to have a single parent. So all the nodes just go to a single parent root node. And we find that even if you do kind of that flat tree structure, TGIF is able to pull out the differences that is actually present in the data. But if you have more information, either based on just measuring the correlation, distance stratified correlation between your high-C data sets, and which ones are more similar than the others, or you want to constrain it based on the gene expression data, you can use that information and specify the tree structure however you would like. We did actually consider figuring out a way to learn the tree structure itself, but that's something that we're still kind of toying around. Hopefully I answered your question. Cool.
So I had a question about your motif enrichment results. So if you could go back to that slide for a second, I just want to make sure I ask it correctly.
This one or this
one? The next one, yes.
Yes.
So you're looking here, this is looking, if I understand it correctly, at like overall motif enrichments. Did you find transcription factors that are enriched whose motifs are enriched at boundaries only at specific time points, but not overall?
Yes, correct. So in this particular case, we are only looking at motif enrichments within day 0. So that is HESC. Now, if we can also apply this if you have either ChIP-seq peaks or atac-seq peaks for any of the subsequent time points, which I'm trying to remember if we have it for that data, for this particular data set, but so this kind of thing is pretty generalizable as long as you have either ChIP-seq peaks or, atac-seq peaks.
Yeah, I'm
But
curious to see if some of these would change. I'm guessing
Exactly.
CTCF would not, but I'm not sure about the others.
Exactly, yeah, so that would be kind of an interesting thing to try out, for sure.
Thanks.
Thank you.
Quick question? Yeah. Just noticing that MYC is in there. So MYC actually tends to be more associated with promoters rather than enhancers in ES cells. So do a lot of your boundaries correspond to transcription start sites or
We
not?
haven't looked into that yet, and there are some studies that show that polymerase, RNA polymerase's elongation action actually enforces a lot of these TADs, so that would be an interesting thing that we would definitely want to look at. Thank you.